Hello everyone, welcome to 10-Minute Physiology. In today's video, I'm going to give you a short explanation as to how cells regulate their volumes. So with that, let's give it a go. So I like to start off by first describing to you the importance of the sodium-potassium pump in maintaining cell volume. So in order to understand that, we're going to look at the ideal cell or a simplified cell model. So this particular cell is submerged in a solution, and this solution has a few different ions. The first is sodium. So the sodium concentration outside the cell is greater than the sodium concentration inside. The opposite is true for a potassium, and the chloride concentration outside the cell is greater than the chloride concentration inside, and then the protein concentration is greater in the inside than it is on the outside. So inside this ideal cell, we have leak channels. So leak channels are channels that are always open, and they allow ions and molecules to pass down their electrochemical gradient. So the first one is going to be the sodium leak channels, which will allow sodium to flow into the cell down its electrochemical gradient. And let's just say 3 sodiums flow into the cell. The next leak channel is going to be the potassium leak channel. So in this leak channel, potassiums are going to move out of the cell, and let's just say we have 2 potassiums moving out. Now inside the plasma membrane, we have the sodium-potassium pump, or the NKP. Now the NKP uses the energy released from the hydrolysis of ATP in order to pump 3 sodiums out and then 2 potassiums into the cell. Now as you see here, the 3 sodiums that came in through the leak channel are all pumped out by the sodium-potassium pump. And all the potassiums that flowed out of the potassium leak channel are brought back in via the pump as well. So what we see here is that the sodium-potassium pump is incredibly important in maintaining the electrical chemical gradients of different ions, in this case sodium and potassium. So the sodium-potassium pump is integral in maintaining the electrochemical gradients of both sodium and potassium. So the next leak channel is going to be the chloride leak channel. And in the ideal cell, the chloride is going to be in equilibrium. So there is no net direction in which the chloride flows. So now what we're going to do is we're going to see what would happen if we inhibited the sodium-potassium pump. So if we were to use a molecule that inhibits the sodium-potassium pump, what would happen is the pump would stop working and different things would happen in the cell. The first thing is that all of the sodium that comes into the cell stays into the cell, and all of the potassium that leaves the cell stays out of the cell. So what you should see here is that 3 sodiums, or 3 positive charges, are coming in, and 2 potassiums, or 2 positive charges, are going out. Therefore, we have 3 positive charges going in, 2 positive charges going out. So this means that in this point in time, the cell has accumulated 1 positive charge. Because remember, we have 3 positive charges through the sodiums coming in and 2 positive charges through the potassiums going out. Now, because of this accumulation of 1 positive charge, this will draw chloride from the extracellular environment into the cell. So we also have chloride now coming into the cell as well. So in summary, we have 3 sodium going in, 1 chloride going in, and 2 potassium going out. Now the sodium, potassium, and chloride ions are all osmotically active particles. So they're all particles. So therefore we can look at each of these individual things as a particle. So for example, the 3 sodiums can be seen as 3 osmotic particles. So if we were to summarize everything, we have 3 particles coming in, 1 particle going in, and 2 particles going out. So therefore, if you were to add all these things together, you would get a net accumulation of 2 particles in the cell. So this means we have an increased number of osmotic particles. And since we have an increased number of osmotic particles, this means we have an increased osmotic pressure. And this means that the cell will draw water from the extracellular environment into the interior. Now, as the cell accumulates water, this will cause the cell to swell. So what we learned from this little experiment is that the sodium-potassium pump basically prevents the cell from swelling. If the sodium-potassium pump were removed or inhibited, What would happen is that the cell would accumulate water, swell, and then it's at a great risk of bursting. So that's why the sodium-potassium pump is very important. So the next thing we're going to look at is how does the cell respond to different changes in volume? So the first thing we're going to look at is what happens, how does a cell respond when it shrinks in size? So the cell will shrink in size when it's exposed to a hyperosmolar environment. So in other words, the solution outside the cell has a greater osmolality than the inside of the cell. And when this happens, the osmotic pressure outside the cell is greater than the osmotic pressure inside the cell, so therefore water leaves the cell and the cell will shrink. So how does the cell respond to this change? Well, this shrinkage is actually going to activate a specific protein called the sodium hydrogen exchanger. So the shrinkage activates this protein, which basically brings one proton out of the cell and one sodium into the cell. Now, as protons are removed from the cell, this causes the pH of the cell to increase. And as the pH increases, this activates another transporter called the chloride bicarbonate exchanger. So what this does is it moves one bicarbonate out of the cell and one chloride in the cell. Now, the sodium that was brought in by the sodium hydrogen exchanger is going to be fed into the sodium potassium pump. And as we bring in more sodium through this exchanger, the activity of the sodium potassium pump increases. So therefore, the sodium potassium pump becomes more active and it brings in more potassium. So potassium is brought in and chloride is brought in, and this is going to be the main mechanism by which the cell increases its osmolality. So in response to shrinkage, the cell is going to increase the accumulation of potassium chloride, which increases the osmolality, which therefore increases the osmotic pressure, which therefore draws water from the extracellular environment into the intracellular environment, and it therefore increases its size. So this process is called regulatory volume increase, or RVI. And there are different types of mechanisms for RVI for different types of cells. For example, one alternative mechanism uses the NKCl, or sodium potassium chloride cotransporter, in order to bring more osmoles into the cell. So what if we did the opposite? So if a cell were put into a hyperosmolar environment, or when we decrease the osmolality outside the cell, what would happen is that the cell would swell. So how does the cell respond to this situation? Well, the swelling can actually activate many different mechanisms. The first mechanism is that the swelling will activate potassium channels. So the potassium channels will allow potassium to flow out of the cell and therefore decrease the osmolality inside the cell, allowing water to leave the cell. Another way that could occur is through the chloride channels. So chloride can leave the cell and do a similar thing, where it decreases the osmolality, decreases the osmotic pressure inside the cell, and therefore allows water to flow out of the cell into the environment. Now, cells can have either the potassium channel or the chloride channel, or some cells may have both of these channels in the mechanism. Another alternative way is through the potassium chloride cotransporter. So the potassium chloride cotransporter co-transports both potassium and chloride out of the cell. So this is another alternative mechanism. So this whole response, all of these different mechanisms, will lead to a decreased osmolality, which decreases the osmotic pressure, which therefore allows water to flow from the cell, and it allows the cell to shrink in size. So this is called regulatory volume decrease, and there are different types of mechanisms for different types of cells, as we've talked about. So this is a summary of what we've talked about so far, both RVI and RVD, and this is basically just a written version of what I just told you. And one thing that I do want to point out is that some of the pathways of RVI inhibit the pathways of RVD and vice versa. So the last thing I want to tackle today is how does the cell respond to long-term hyperosmolality? So in general, when a cell is exposed to long-term hyperosmolality, the cell is going to accumulate something called osmolytes. So osmolytes are impermeant alcohol derivatives of common sugars as well as two different amines. So of the sugars, sorbitol and inositol, and then we also have the two amines listed here. So how does the cell actually do this? So if the cell is exposed to a long-term hyperosmolar environment, the cell will shrink, of course, and in response to this shrinking, the cell is going to increase the synthesis of a specific enzyme called aldose reductase. So aldose reductase is an enzyme that converts glucose into sorbitol. Now remember, sorbitol is impermeant or impermeable to the cell membrane. Therefore, sorbitol remains in the cytosol and it acts as an osmotic particle, so it helps draw water from the surrounding environment into the cell. Another mechanism is that the shrinkage will also increase the synthesis of specific co-transporters. These co-transporters are going to co-transport sodium and other particles into the cell and allow the cell to increase its osmolality and draw more water in. And some things that it can bring in through these co-transporters include inositol, taurine, and other amines. So that's it for this video. I hope it helped you understand how the cell responds to different volume changes and how the cell maintains its volume. I hope to see you in the next video, and thank you for watching.